Today, I want to tell you a story about my mother. For years, my mother dreamt of making the perfect cheesecake. She wanted to make a tall and fluffy cheesecake, but every time she tried, she got an unexpected result. Her cake kept on collapsing, and she would not understand why. One day, we asked her, "Mom, did you follow the exact recipe for the cheesecake?" And she replied, "Yes, of course. All I did was to reduce a bit of sugar." What my mother did not understand is that the reason her cake kept on collapsing is that she reduced the amount of sugar. She knew what ingredient she put in and what the final outcome is, but she did not understand the process in the middle. In genetics, we often face a very similar problem. We have our genes, a recipe. They have a sequence that encodes all of our traits. It could be physical traits, like eye color or height, but it can also be more complex traits, like cognitive abilities and even behavior. And sometimes we have a small change in the recipe, a mutation, and this leads to a change in our traits, but we don't always understand why. So, we have genetic diversity. Each one of us has a slightly different DNA sequence, and this leads to a diversity in traits. We can see this diversity all over the living world. For example, a small change in the DNA sequence of the cat explains why some cats have pointy ears while others have round ears. A small change in our DNA sequence explains why some of us really like cilantro and others think it tastes like soap. Today, I want to talk to you about the same phenomena in the context of autism spectrum disorder. Autism is characterized by difficulties in social communication and strict repetitive behavioral pattern. Autism is diagnosed using a psychological assessment in the clinic. However, for many years, we know that autism also has a biological aspect. For example, we know that people diagnosed with autism tend to have an abnormal brain structure. More so, we know that autism has a strong genetic basis. It's important to understand that although we know there is a biological aspect, the biological mechanism underlying the disorder is still poorly understood. So you might be wondering, how do we even know autism has a genetic basis? Well, there are two main ways. The first one are twin studies. So if we look at twins, which can be either identical or non-identical, meaning they share either 100% or 50% of their DNA sequence, and we focus on cases where one of the siblings has autism, we can ask, what is the probability the other sibling will also have autism? And when we do it, we find out that this probability is very, very high. More so, we can identify what we call autism-associated genes. So if we take a lot of people diagnosed with autism and sequence their DNA and compare it to people that are not diagnosed with autism, we can identify specific genes that tend to have mutations in people with autism. So, we have these autism-associated genes, which again are genes that tend to have mutations in people with autism. We also have abnormal brain structure. More precisely, we know that people with autism tend to have an abnormal brain size. So they can have either a bigger than average brain size or a smaller than average brain size. And we can even connect the two. So we can say that specific genes tend to be more associated with the bigger brain size, while others with the smaller brain size. But what we don't understand is the process in the middle. We have some sort of a black box. Why are those mutations affecting the brain size? Now, why is it a black box? In order to understand how mutations that exist from the very early stages of development eventually affect the brain size, we need to be able to look at brain development. But brain development happens mostly throughout embryonic development. Here you can see photos of the embryonic brain. And you can see that the brain starts to evolve as early as week 4 of the pregnancy. And by the late stages of the embryo, it is already well developed. So, we want to understand how mutations in autism-associated genes affect the early stages of brain development. But how can we do it? Since we don't have access to the brain, and the embryo, we need some sort of a model. In our lab, we are using embryonic stem cells. These are cells that derived from the early stages of the embryo, and we can grow in the lab. Given the right conditions, these cells can develop, can differentiate into any type of cells in our body. They can become brain cells, they can become heart cells, and they can become liver cells. So we are taking those embryonic stem cells, and we are generating by ourselves mutations in the autism-associated genes, allow the cells to become brain cells, and ask what happens. Now, that might sound relatively simple, but you need to keep in mind that the brain is a very complex organ. Here, you can see a real photo of the brain, where each color represents a different type of cells. And you can see we have many different types of cells in our brain. For example, we have our neurons, which are the main cell type of the brain. They communicate with each other using electrical signals. But we also have other cell types in the brain. Oligodendrocytes, for example, are an important supporting cell type that is critical for brain function. Going back to our experiment, we are not only asking whether our cells can become brain cells upon mutation, we are also asking what kind of cells are generated and in what proportion. So, can we use this model to better understand this black box in the middle? We found that the same genes that tend to be associated with bigger brain size also tend to accelerate the rate of the differentiation of the cells. Specifically, these cells tend to generate neurons much more earlier than expected. On the contrary, the genes that are associated with smaller brain size also accelerate the rate of the differentiation, but toward a different direction. These cells generate oligodendrocyte, another cell type, much more earlier than expected. Our results shed some light on this black box that we talked about, but they also raise follow-up questions, or more black boxes. For example, how is it that changing the rate of the differentiation affect the brain size? More so, how are those mutations actually affect the rate of the differentiation? This is the path we are currently studying in the lab. I want to conclude by saying that this study, together with many others, is aiming to add a small piece to the puzzle. And we believe that by joining all of those small pieces together, we will be able to better understand what autism is and maybe offer better diagnostic and therapy in the future. Thank you, and I would be happy to take your questions.